We have loaded the plasticware and the reagents into the SP100. Now we will load the samples.
Ensure that you have planned an order for your samples, recording the correspondence between Seer sample tube numbers 1 to 20 or 1 to 40 listed on the barcode labels, and plasma sample numbers or other sample identifiers. Confirm the centrifuge has reached 4 degrees Celsius. Then centrifuge the samples at 5000 G for 2 minutes. For each sample, transfer 240 microliters of plasma into the appropriate Seer sample tube. Minimize aspiration of residues that might settle at the bottom of the tubes. Avoid aspirating any lipids that collect at the surface of the samples. Minimize bubbles forming at the surface of the samples. Cap the sample tubes, then centrifuge at 5000 G for 1 minute. Set aside the sample tubes on ice or at 4 degrees Celsius. Remove the tube carrier 1 from the work deck. Load each sample tube into the tube carrier according to the layout shown on screen. Load tubes from the back to the front, beginning with slot 1. Be careful to load the sample tubes in the precise numerical order. Failure to load tubes in the correct order will result in an error during barcode scanning. Place the plasma control and MPE control tubes into the tube carriers according to the layout shown on screen. Load tube carrier 1 onto the work deck.
Next, we will load the nanoparticles.
Remove tube carrier 2 from the work deck. For each of the 4 nanoparticle tubes, tap the tube on the table to ensure that the lyophilized beads are settled and none are stuck to the cap. Uncap the tube, then place it into the tube carrier according to the layout shown on screen. Load tube carrier 2 onto the work deck. Verify that all checkboxes are selected on screen. Then select Continue.
Next, we will complete the setup and start the method.
At the labware check screen, verify the correct placement of each labware item on the work deck according to the layout shown on screen. As you verify each item, select its corresponding checkbox on screen. Select Continue to advance. At the sample name screen, enter the name or identification number of each sample. To populate all sample name fields with values from a file, select Load Sample Names. To populate all sample name fields with default values, select Default Sample Names. Select Continue to advance. At the Begin Labware barcoding screen, confirm that barcodes are fully visible. Orient all labware such that barcodes are facing the barcode scanner. Confirm that each carrier is pushed forward against the stop hooks at the back of the auto-load tray. Select Continue to advance. Close the front protective cover. Select Continue. Barcode scanning will begin. At the Setup Complete screen, select Run to start the method. The ICS locks the front protective cover. The method takes approximately 6.5 hours to complete. During the method, the ICS displays each stage with time estimates.
We started the method. Next, the run will end and we will unload the work deck.